Welcome to another OmniFocus Fitness video. Today we will be discussing glycogenolysis, which is the brother of glycogenesis. I already have a video on that, but to complete this series we will need to understand how glycogenolysis works. You will know everything you need to know about glycogenolysis. I will break down the chemical structure, I will go through all the biochemistry, and we will go from where we left off in the glycogenesis video, and we will loop back around to the end position of glycogenolysis. So everything will be covered. In one video, you will learn all that you need to know. Are you ready? 3, 2, 1. Let's go. So first things first, of course, we need a definition. What is glycogenolysis? Glycogenolysis, let's keep it simple, is basically just the breakdown of glycogen into glucose. Leave it at that. You will know more details once we go into the chemistry of it all, but for now, just know that. That's a good definition. That's very basic, and it's very true. Where does it occur? Well, we know that glycogenesis ended up in the cytoplasm, and that is no different for glycogenolysis because, again, we're going to be picking up right where we left off with glycogenesis, the end of the glycogenesis video. So the glycogenolysis occurs in the cytoplasm of primarily the liver cells and the muscle cells. So I already mentioned that Excuse me for a second. Let's start that over. I'm not even going to cut that out. Let's just start over. We already know that glycogenolysis is the brother of glycogenesis. And why does it occur? Well, glycogenesis, if glycogenesis is the building of glycogen out of glucose, it's used as a storage system for quick release energy, and glycogenolysis allows for that quick release of energy. Now, at the end of this video, you will find out exactly how glycogenolysis, again, on a very chemical level, how it impacts your body and how that breakdown of glycogen can really be helpful. But from a macro perspective, anytime you're going to be doing any sort of high intensity exercise, if you're got your adrenaline running, things like that, in those cases your body's going to be trying to use glucose, and it's going to be getting that glucose from your glycogen stores. As such, your body always goes through glycogenolysis to get those stores of glucose, so go from a stored form of glucose to a viable form of glucose for your body to actually use. And again, it uses glycogenolysis to do that. So let's figure out how. If you haven't seen my glycogenesis video, then you will not know that now is the portion where I switch to a blackboard method, and we will explain things from a more visual perspective. So let's go ahead and do that, and this is the last time you're going to see my face. Might be a good thing, but this is the last time you're going to see my face. So I will see you in the next video, and enjoy the next portion. Hopefully you enjoy it. If you're a nerd like me, then you will enjoy it. Anyways, I will see you in the next video. Peace. Welcome to the far more in-depth part of this video. Okay, so the first step that I kind of want to look at, like to look at things from a greater perspective and then move into the details. So let's go ahead and do that. Let's get a little bit of context. If we look over here, we can see that there's a very simplistic cell. So that's supposed to be a cell, at least. And at the end of the glycogenesis video, we ended up inside of the cell. So we were in the cytoplasm, which, as we know, is where glycogen is held. So glycogen is activated glucose molecules, and in glycogenolysis, this topic, what we're trying to do is trying to move activated glucose, or I guess move glucose that's been stored as glycogen back out of the cell, or at least that's what we're going to go with for now. So we are talking about moving glycogen, which is again activated glucose, to glucose, free glucose. And to give this a little bit better picturesque style to it, imagine these red dots being glycogen, and then imagine these yellow dots being glucose. So what we're trying to do is simply move from inside of the cell back out. So again, glycogen to glucose. So that's the overall step. That's what we're trying to do, and now we will go into the details of how exactly that occurs. Let's go ahead and do that. Okay, so here we are in step 1. What we're trying to do is move from UDP glucose to glucose 1-phosphate. If you're familiar with glycogenesis, you're familiar that UDP glucose is activated glucose. Activated glucose is the glucose residues that make up glycogen branches as well as chains. So the branches here as well as the chains. This is supposed to represent a glycogen chain / branch. And we're in a position now with glycogenolysis that we have to now break these branches and these chains down into smaller forms. So to move in the reverse direction of what glycogenesis did for us. So as far as building glycogen, now we're trying to break that back down. And if you're familiar with glycogenesis, you're familiar that there are two different enzymes that play a role on building the glycogen. Well, the same is true of then breaking down that glycogen, and both of these enzymes are very telling, specifically towards the bonds that pull together these activated glucose residues. So to explain that a little bit further, you might be familiar with the idea of these they're called alpha 1G4 glycosidic bonds, and they stand between each glucose residue except for one spot. And that one area is the intersection between a branch and a glycogen chain. So detailed here, they would be located right about here. You're going to have to forgive my drawing abilities. They still haven't improved since the last video. So these alpha 1G4 glycosidic bonds, as well as the alpha 1G6 glycosidic bonds, have been put together in a very concise, rule-based system. And for us to then break apart these bonds, we need, as I mentioned before, two enzymes. So let's go ahead and talk about the first one. Okay, so the first enzyme that we're going to look at is glycogen phosphorylase. And glycogen phosphorylase acts on the alpha 1G4 glycosidic bonds. So it's going to come in here and break these bonds and going to also phosphorylate these glucoses. So you'll notice that I'm now changing it to orange because we now have phosphorylated glucose molecules. So glucose 1-phosphate. I'll just go ahead and put a P here. I might not be able to. Okay, well, that's good enough. Kind of poor job, but you get the point. As these break, they move from here outwards. Okay, so we got glucose 1-phosphate. They're free to a degree. They have a phosphate attached to them for the following step following this one. Now, glycogen phosphorylase has to follow the same type of rule that glycogen synthase followed in glycogenesis. And in this case, as it breaks these bonds, it can only break these alpha-1G4 glycosidic bonds up to 4 residues away from the main chain. So on a branch, so we're 1, 2, 3, 4 residues away, so it can still break this one. And these all get turned into glucose 1-phosphate. So that is literally the extent of glycogen phosphorylase, to break these 1G4 glycosidic bonds up to 4 residues away. So 1, 2, 3, 4. Those are terrible numbers. 4 residues away from the main chain of glycogen. And then just phosphorylate these again, to phosphorylate these glucose molecules to make them glucose 1-phosphate. And that's it. That's all that glycogen phosphorylase does. And now is where another enzyme, the following enzyme, then takes over and does kind of a cleanup process, as well as moves the reaction forward some more. Okay, so we know that glycogen phosphorylase is now done and can no longer continue. So what happens? We still have 4 residues located right here. We still have 4 residues that are attached to the chain, making up a very small branch. Well, as I mentioned before, there's a second enzyme that comes in, and that second enzyme, which is kind of fitting because, again, going back to glycogenesis, we were talking about a branching enzyme, and this one is called the debranching enzyme. And now the debranching enzyme not only plays on alpha-1G4 glycosidic bonds, but also plays on the alpha-1G6 glycosidic bonds, but does so in 2 very distinct ways. And these 2 ways are possible due to 2 activation sites on the debranching enzyme. And those 2 activation sites are called glucosyltransferase and glucosidase, which sound complex, and really they are pretty complex names, but let's break down what each does. So what does each active site do on debranching enzyme? Well, the first one, glucosyltransferase, if you'll focus actually just on the word transferase, what does that sound like? The transferring, so the word transferring, so movement from one place to another. And that's exactly what this active site actually does. What it'll do is it'll take these 1, 2, and 3 glucose residues, and it will cleave here. So grab these 3, and what it'll actually do is it will add these 3 residues to a different branch. So effectively removing these. So these don't exist here anymore because they've moved up here and have been added to a different branch. And that leaves us with one final residue. And this residue is acted upon by the glucosidase activation site. And all that does, it's very simple, is it breaks this 1G6 glycosidic bond. And when it breaks said bond, what happens to this final glucose residue is it becomes a free glucose. So you'll notice that I do not paint it in orange, and that's because it actually does not become a glucose 1-phosphate. While most of the activated glucose that their chains are broken, they eventually get turned into glucose 1-phosphate and go through the following steps that we're going to discuss. In this case, what glucosidase does is it does not add a phosphate like glycogen phosphorylase did. Instead, it just breaks that bond, and that glucose is then a free glucose molecule. Okay, so we've covered all the information, but I tend to like to give several different visuals of the same information. If you feel like you've got it, please move on. But real quick, what I'd like to do is move down and give you 2 different visuals with a little more specific. So if we're looking at the actual chemical structure of glucose in this case. So what we're talking about here is glycogen phosphorylase, as can be seen right here, which is again our first enzyme, and we've got 2 glucose molecules that are stuck together. So this is glycogen, obviously. Again, we know activated glucose makes glycogen, so we've got several different, and granted there are probably many more glucose molecules that are stuck this way, but it just shows 2. And glycogen phosphorylase comes in here and adds a phosphate as well as breaking this bond. So as you can see here, we have, this is glucose 1-phosphate because that phosphate was added to the 1 location. You can see again here phosphate, and here a phosphate, which is added to this 1 location here. And this, if you'll just, I mean, you can plainly look at this, just the structure. I mean, looking at this box, if I can draw correctly, let's try that one more time. Alright, so this box, if you look at this box and simply compare it to either one of these glucose residues or molecules, you'll see that they're identical. So this glycogen phosphorylase is only phosphorylated 1, and it only does this 1 at a time. So that's how glycogen phosphorylase then cleaves a bond and adds a phosphate to make glucose 1-phosphate. And as we know, the majority of these residues that are cleaved end up as glucose 1-phosphate. So let's go ahead and move down here and look at what debranching enzyme does. So again, we know that here we have a perfectly simple diagram of glucose residues. So here we've got 4 residues, and as we know, yet again, that glycogen phosphorylase stops once it is 4 residues away from the main glycogen chain. So here it is, it's stopped. Glycogen phosphorylase no longer moves on, and debranching enzyme comes in here. So debranching enzyme will come in and cleave this bond and then move this over here. So you can see it's no longer, there are no 3 residues here, it's moved them to a different branch. Having said that, and that's again through the transferase mechanism on debranching enzyme. So then if we move over here, we can see that debranching enzyme uses the glucosidase activity system so that it can get rid of this 1. So it breaks this bond here, so it cleaves this bond. It does this through, it's assumed, we're thinking, I'm pretty sure that we're not 100% certain on this, but it's assumed that it's through hydrolysis that this occurs. So H2O, water, is added to the reaction, which then breaks this this bond. And then we, because we're using hydrolysis, we're not adding a phosphate. This becomes a free glucose. So again, this becomes a free glucose and just floats off. So again, there's nothing here to signify that that residue stays. And that's that. You know, again, pretty simple. It's pretty interesting, but it's you know, if we just go over it very simply, we can really understand how all this stuff works. So that's that. That's the first step. That's all we need to know, and we can go ahead and move on to the next step. See you there. Okie dokie, moving on to step 2, we are trying to move from glucose 1-phosphate to glucose 6-phosphate, and this should be a spitting image of what happens in glycogenesis. In that case, we basically have the reverse. We have glucose 6-phosphate moving to glucose 1-phosphate, but the mechanism by which this happens is the exact same for glycogenolysis. So let's go ahead and label this stuff. Okay, we've got this labeled. Right here we have our glucose 1-phosphate, and over here we have our glucose 6-phosphate. Now, before we talk about this mystery molecule here, I would like to quick kind of break down the name. So when we're talking about free glucose, we're talking about just glucose here. So these names are pretty intuitive, and it helps to know how to go about understanding these names so that we can understand specifically in this step how everything's outlined. If you can understand that, it's really easy. So glucose by itself would just be called glucose, and here we have the number 1 and phosphate. So as we know, in the step 1 we had glucose, which had a phosphate attached to it once the 1G4 glycosidic bond was broken, and we ended up with glucose 1-phosphate. Now the 1, so the phosphate's accounted for and the glucose is accounted for, so we have a molecule glucose and we have a phosphate that was added to it, and the 1 signifies where it was added. So in the first position of the glucose molecule, because there are several different positions that a phosphate could go, and in this case it was placed on the 1 position. So that's what's signified here. Imagine this being glucose, this orange being a glucose molecule, and it's added a P at the top because it is in the first location. Then let's go ahead and quick skip over here. The exact same thing. So think of the exact same mentality. You have a glucose molecule, which is in this darkened circle, and you have a phosphate, so we still have a phosphate attached to it, and a 6, which signifies, I'm sure you can guess, the 6th location on the glucose molecule, and that's why we've moved it to down. So instead of being located up here like it is here, it's located down there. So now let's go ahead and discuss what this mystery molecule in the middle is. And we can do that by looking at the name, and that's the whole reason why I introduced you to the names. Here we are looking at glucose 1,6-biphosphate. So again, glucose, the colored in circle, 1, which 1 over here was at the top, so we've got him at the top here again, and a 6, which is just like here at the bottom. So we've got a phosphate in both locations, and because it is both, we are looking at biphosphate 2. Okay, pretty simple. How does this occur, however? This all occurs with the help of an enzyme called phosphoglucomutase. And this enzyme, what it does is, because we already have a phosphate, it itself has a phosphate on it. And when it comes in contact, it interacts right here and right here. So the first thing that it does is it adds a phosphate to the 6th location, which then gives us glucose 1,6-biphosphate. Now then it is, at this point, it is dephosphorylated. So the enzyme is dephosphorylated. It no longer has a phosphate attached to it because it has given that phosphate to the glucose 1-phosphate to create glucose 1,6-biphosphate. And at this point, then the enzyme then reorients itself. And this is all right here. So it reorients itself and then ends up in this situation right here where it reacts again with the glucose molecule, which in this case is set up as a glucose 1,6 biphosphate molecule. And at this point, after it has reoriented itself, it rephosphorylates itself. So the glucose molecule then loses one of its phosphates, so it's no longer a bi because it loses this one here. So it loses this one, no longer becomes a bi, and we are left with glucose 6-phosphate, which is right here. And that is the entire step. Okay, so that was the entire step. But again, I would like to go ahead and go through and go over it one more time by looking at the chemical structure. And I apologize for the size of this picture. I couldn't really find one that wouldn't get really blurry when you blew it up. So this is the best I can do. But we are moving in this direction in the way that we read it. And this is the exact same. Okay, so we've got our phosphoglucomutase located right here with a phosphate, and we already have a phosphate right here. So the glucose 1-phosphate already has a phosphate at the 1 position. Then what happens is it interacts here, and you can see that this phosphate no longer exists here. It's no longer attached to the phosphoglucomutase and has now been positioned here. So we have a phosphate here and we have a phosphate here, the PO3. And that gives us glucose 1,6 biphosphate, as I've already explained. And then the phosphoglucomutase then reorients itself, and it acts here again by taking away this phosphate. So this phosphate then disappears. You don't see it here, and where did it go? That's because it is up here. So the phosphoglucomutase has then rephosphorylated itself and has left only one phosphate on the glucose, and it is on the 6th location, so the glucose 6-phosphate. That's that, folks. That is it. That's all that there is for step 2, and it is very, very similar to what happens in glycogenesis. So let's go ahead and move to step 3, where things get actually pretty interesting. Alright, so here we are, and we are in basically our final step. And in this final step, we are going to really realize what exactly glycogenolysis can do for us on a cellular, a molecular, a chemical level. And you'll see here that I've got listed 3 different systems, and it's glucose 6-phosphate moving to various energy systems which are dependent on what the body needs. And I'm not going to go into a massive amount of detail, but give a little bit of a backstory would be helpful to really understand why glycogenolysis is so important and how it affects us. Well, in the first case, glycolysis, which is something that our body uses all the time, which honestly all 3 of these our body uses quite a bit, but glycolysis is a huge one because glycolysis ends up giving us energy in one way or another. So again, in every single step in each of these 3 energy systems, we're starting with glucose 6-phosphate. And specifically in glycolysis, the use of glucose 6-phosphate is to move down the glycolysis chain, which goes through tons of different reactions, which we won't go into. But the first one, just to get us kind of jump started, if you were to look at like a map of glycolysis, you would understand that this very first step is kind of the one of the beginning steps to the whole glycolysis system. And this is started by an enzyme called phosphoglucose isomerase. And again, I'm not going to go into a ton of detail, but eventually that turns into the molecule fructose 6-phosphate. And you can look at a glycolysis article or video to get your bearings straight, but that should let you know where we are at that point. And as far as where glycolysis occurs, just real quick, if we were to move down and look back at our cell, this is again the cytoplasm where all of this has occurred so far. Glycolysis also occurs within the cell. So moving back up, then we can look at the release of glucose. And what exactly do I mean by that? Well, what's meant by that is that it is possible for glycolysis, in this instance, glycolysis is using energy immediately, so using that glucose 6-phosphate to create energy immediately. While the release of glucose, it is possible for certain cells, specifically liver cells and not muscle cells. So this is only applicable to liver cells, that they release glucose back out of themselves, out of the cell, and into the bloodstream to then conserve all kinds of purposes, but usually to then supply glucose for other cells downstream from in the blood system. In this case, we are going to actually end up with free glucose, so just a plain glucose molecule. Let me make that more of a C. Glucose plus and then what's going to detach from that due to hydrolysis. So again, this is becoming hydrolyzed so that we can get rid of that phosphate on the 6th location of glucose. So this position right here, remember there's a phosphate on the 6th position. Well, we have to get rid of that, but we don't want to add a phosphate to it. Nor do we want the enzyme to necessarily take the phosphate. So what we use is hydrolysis, so the addition of water to then break the phosphate off of the glucose and have it be individual. So we have the phosphate individual of the glucose molecule. And because of that, if we go down here, we can see that this is an instance in which the glucose then moves out. Hopefully you can see that. Out of the cell. So that's a big yellow arrow. So from the inside of the cell, which again everything so far has been inside of the cell, we're moving that back out of the cell into the extracellular fluid, into the blood. And finally, if we look at the third energy system, which is a unique one, is called the pentose phosphate pathway. And this pathway actually creates something called NADPH. And it's not 100% accurate, but for the sake of this video, we're going to keep things pretty simple. And the pentose phosphate pathway is actually useful for different anabolic processes as well as aiding in immune defense. If you want to know more about that in the future, you can be rest assured I'll cover it, or just look it up somewhere else. But that's all you need to know for now, for simplicity's sake. So we've got these three energy systems that use glucose 6-phosphate. And again, glycolysis is for immediate energy, right away, right then, going down the glycolysis pathway. The release of glucose, which can only be done by the liver, releases glucose by hydrolyzing the glucose 6-phosphate, getting rid of the phosphate, and then just having the glucose molecule and pumping that back out into the blood system for other cells downstream to use. And the pentose phosphate pathway, which I just said helps with immune defense as well as several different anabolic processes, and does a few other things which I won't mention here. So that's that. Alright, now let's go ahead and look at this one more time, but with different pictures. Okay, so this might look a little overwhelming, but don't worry, we have covered almost everything on here already. Everything that you need to know, at least, to understand glycogenolysis. So let's quick look at, let's go in order. Let's look at glycolysis. So as we know, here we have our trusty glucose 6-phosphate that we were just talking about, and this is where glycogenolysis would come in. So you can get glucose 6-phosphate from either a free glucose coming from the outside being transported into the cell, and then a particular enzyme called hexokinase, which you can find out about in the Glycogenesis article and video. You can get glucose 6-phosphate that way, or you can get glucose 6-phosphate the way that we just described, which was through glycogenolysis. And then here we have our phosphoglucose isomerase, which I just discussed, then acts on glucose 6-phosphate to turn it into fructose 6-phosphate. And then all these other steps are the rest of glycolysis. You don't need to know any of this stuff just to understand. I'm just trying to give you a placement of where all this stuff fits in. So it's between 1 and position 2 of glycolysis. So let's go ahead and move on to the glucose release. Releasing glucose, on the other hand, there is a little bit more information that I'd like to cover in this subject. So here we have our cell, and you know, cells aren't always perfectly round, but for the purpose of this, we'll just imagine that this cell is perfectly round. It doesn't really matter. So here we have the outside of the cell, and here we have the inside of the cell separated by the cell membrane. And many things have to move inside and outside of the cell through something called facilitated diffusion. And facilitated diffusion is basically using a protein to move across the cell membrane, because the cell membrane gives that distinction between inside and outside of the cell, and it is extremely important, otherwise the cell would just not exist. So it gives it its structure, keeps things in, keeps things out, regulates what goes in and what goes out. Well, what we just talked about was the release of glucose. So in this diagram, you can already see right here glucose. So glucose molecules don't just suddenly just appear outside. They actually use what's called a GLUT2 glucose transport protein. And as I just explained, transport proteins allow that diffusion across the cell. So it's spelled like this, a GLUT2 transport protein. And you can just imagine a protein that's embedded into the cell membrane like that, allowing glucose to then escape and go outside into the extracellular liquid. So that's really the only other piece of information that I wanted to quick add, and I thought that it would be best to just wait till you actually had a better image. So let's go ahead and move on to the pentose phosphate pathway, and this one obviously again looks kind of complex, kind of like what glycolysis looked like. But if we just break this down in the stuff that we just real quick covered, again here we have glucose 6-phosphate. And our glucose 6-phosphate then goes in, oh well, here you can see that it goes into glycolysis, just like over here, right? But it can also move and get acted upon by glucose 6-phosphate dehydrogenase, which then creates our NADPH, which was written up here. And that's all you need to know. You don't need to know anything else. If you want to know more about this system, then watch a video or article on the system. But for the time being, that's all you need to know. It just gives you your bearings so that you understand how glycogenolysis fits into the equation with all these three different energy systems. And that is that, folks. That is it. That is all that is in this video. It is quite long. These tend to be quite long, and my videos in general tend to be quite long, but hopefully it was informative. I really hope that I explained it well enough for you, and if you have any questions, feel free to ask me. And any details, there are many details that I did not quite go over. Those will be located in the article, which is linked below, or it will pop up on the screen momentarily. If you thought the information was informative, please subscribe. Definitely helps me and it motivates me to keep pumping out these videos. Things will remain free, and thanks for tuning in. Catch you next time.